Assalamu alaikum, beautiful people. Today we will discuss about the mitochondria, the energy currency of the cell. Here we discuss about history, morphology, mitochondrial vesicles, and function of mitochondria. Combination of two words, mitos means thread-like and chondrion means granule or vesicle. Thread-like membranous organelles found in eukaryotes, discovered by Koltiker in 1850. Altman named it bioblast in 1890. Bender named it as mitochondria in 1897. Killen and Cover in 1940 found that it is the central part of cell in oxidoreductive reactions having enzymes and coenzymes for electron transport, chain, citric acid cycle, and chemiosmosis. Their main job is energy conversion, the oxidized glucose to produce energy that the cell can use. The process is called cellular respiration, which makes ATP a molecule which the cell can use as a source of energy. In addition to supplying cellular energy, mitochondria are involved in a range of other processes such as signaling, cellular differentiation, cell death, cell division, and cell growth. Mitochondrion, Greek mitos means thread and chondrion means granule. Morphology. Morph means shape and logi means study. Size 0.2 to 0.8 micrometer in width or 0.5 to 7 micrometer in length. A mitochondrion contains outer and inner membrane. These are made up of phospholipids, double layers, and proteins. The two membranes have different properties. Because of this double membrane organization, there are five distinct features within the mitochondria. They are the outer mitochondrial membrane, the intermembrane space, the space between the outer and inner membrane, the inner mitochondrial membrane, the crystal space formed by unfolding of inner membrane, and the matrix space between the inner membrane. Here in the diagram, all the structures are shown. Outer membrane is smooth, working as a boundary between cytoplasm and mitochondria. It is considered as homologous structures with protein coat present around the bacterial cell membrane. It contains porins like proteins permeable to NAD, NADP, ATP, ADP, and coenzyme A. Inner membrane is folded in finger-like projections directed inwardly, known as mitochondrial crest or cristae, which extends into the interior or a matrix of the organelle. Inner membrane representing the major working compartments of mitochondria. The composition of the intermembrane space is similar to the cytosol with respect to ions and small molecules. Consequently, the inner mitochondrial membrane is the functional barrier to the passage of small molecules between the cytosol and the matrix and maintains the proton gradient that drives oxidative phosphorylation. Matrix contains the mitochondrial genetic system, ribosomes, as well as the enzymes responsible for the central reactions of oxidative metabolism. Clear? The oxidative breakdown of glucose and fatty acid is the principal source of metabolic energy in animal cells. Here you can say the question that is ever asked, where the energy is formed. Energy is actually formed after the breaking of glucose and fatty acids. The initial stages of glucose metabolism, glycolysis, occur in the cytosol where glucose is converted into pyruvate. Pyruvate is then transported into mitochondria where its complete oxidation occurs to carbon dioxide yields the bulk of usable energy obtained from glucose metabolism. This involves the initial oxidation of pyruvate to acetyl coenzyme A, which is the broken down to carbon dioxide via the citric acid cycle. The oxidation of fatty acid also yields acetyl coenzyme A, which is similarly metabolized by the citric acid cycle in mitochondria. The enzymes of the citric acid cycle located in the matrix of mitochondria thus are central players in the oxidative breakdown of both carbohydrate and fatty acids. Hope you understand. Number of mitochondria in a cell. The number of mitochondria present in a cell depends upon the metabolic requirement of the cell and may range from a single large mitochondrion to thousands of the organelles. Metabolically active cells have greater number of mitochondria, normally ranges from 500 to 1000 per cell. A frog egg may contain up to 10,000 per cell. Mitochondrial vesicles. In 1960, E. Colliker observed small lollipop like structures in inner membrane towards the mitochondrial matrix, named them as mitochondrial particles. These are composed of enzymes assembly required for oxidative phosphorylation to obtain energy. These can be isolated by chemicals, e.g., digestion, sonication using high frequency sound waves. It acts as ATP synthesizing complex. It comprises of three parts: F1 particles, head, submitochondrial particles or coupling factors, F node or stalk, or third is membrane piece. F1 particle. F1 particle, rounded part of mitochondrial particle. It contains 10 molecules of four different kinds. They all work as ATPs. Okay, what is stalk? It forms the neck of the mitochondrial particle. It connects F1 to membrane piece. It is made up of a regulatory protein called the oligomycin, sensitive regulatory protein. It regulates thermogenesis, temperature regulation. It is sensitive to following antibiotics: oligomycin, that acts as an inhibitor of OSRP by inhibiting the phosphorylation only, but electron transport chain goes on. 2,4 dinitrophenol. It is also an inhibitor of OSRP by blocking the activity of phosphorylation. Rotamycin, that inhibits both electron transport chain and phosphorylation. Okay, move towards the membrane piece. The part present across the inner membrane lies in intermembrane space called the membrane base. It acts as a proton pump that transfers proton from inner membranous intermembranous space to matrix, produce a proton motive force that helps in phosphorylation. It also contains OSRPs. Function of mitochondria. Its function is oxidative metabolism, protein synthesis, self-replicating organelle, and fatty acid oxidation. Oxidative metabolism. Mitochondria is the powerhouse of the cell. It produces large amount of energy by oxidative metabolism of food. By generating large amount of ATP, acetyl coenzyme A, formed in cytosol, enters into mitochondria. Outer membrane is highly permeable, but inner membrane is impermeable to this complex. It contains carnitine shunts for its transport. Acetyl coenzyme A enters into the Krebs cycle and generates a large amount of energy through oxidative phosphorylation. Move towards the protein synthesis. Mitochondria has its own ribosome smaller than that of prokaryotic and eukaryotic ribosomes. So it is capable of synthesizing its own polypeptides. Self-replicating organelle. Presence of DNA and ribosome in mitochondria makes it semi-autonomous. Self-replicating organelle. It does not depend upon cell for its reproduction. Fatty acid oxidation. Lipids and fatty acids are highly energy compounds as compared to carbohydrates and proteins due to high ratio of carbon-hydrogen bonds. A mole of fatty acids can release 9.2 calories of energy as compared to proteins and carbohydrates. Mitochondria also contain enzymes for fatty acid oxidation. Fatty acids cannot enter into mitochondria directly, but in the form of acetyl coenzyme A. Fatty acids are oxidized by beta oxidation in mitochondria. Hope you enjoyed the video. Stay motivated. God bless you. Allah Hafiz.